Okay, so this is where we left off last time. And let's just put things into kind of context again, because this really is a incredibly remarkable system, and it really gets to the heart of who you are as a person and what makes you you. These are really what make you you, the proteins that you produce. Many times they're enzymes, but sometimes they're other structural proteins or transport proteins. But proteins really determine the characteristics of cells, and cells determine the characteristic of organisms. And so these are responsible for determining your phenotype. Okay? But how do we know how to make these particular proteins? And why do different people have different abilities to make different proteins? Because you have different genetic information. You have differences in your genes. So this message reflects the information that was encoded into the DNA molecule by the process of transcription. DNA was copied into a messenger RNA. Other genes were transcribed into the important ribosomal RNAs, which are important functional parts of this machine, the ribosome, that's involved in translation. And other genes encode these transfer RNA molecules, which are responsible for grabbing amino acids and eventually joining them together to form polypeptide chains. The information that tells the kind and order of these amino acids, or the primary structure of the polypeptide chain, is encoded into this message. Okay? So there's some really interesting biochemistry and kind of nanotechnology almost operating within your cells to determine their particular characteristics. So, we've got the main players now. We've got the message with the code words that specify particular amino acids. Please make sure that you understand that code words specify amino acids. They do not specify proteins. Okay, proteins are chains of amino acids, but a code word does not specify a protein. A lot of you were saying that in your essay questions. Codons code for amino acids. Okay? We've got the transfer RNAs, whose anticodon will base pair with a complementary codon on these binding sites on the ribosomes. And the transfer RNA will have been loaded up or charged with a particular amino acid. Enzymes, the amino ACLT RNA synthetases, help to ensure that the proper amino acid gets stuck onto the proper transfer RNA. And then this is just a matter of basically reading these code words and stringing together the amino acids to form the polypeptide chain. The ribosome moves down the message in the 5' to 3' direction, or actually probably more realistically, the message slides through the ribosome, but effectively this message is being read in the 5' to 3' direction. So that these represent code words that have not yet been read in the message, and these represent code words that have been read, and they're represented by those amino acids that have been strung together. So, in the process of translation, we've got the code words being read by the ribosome/tRNAs. And the result of this is a functional polypeptide chain, which will have some particular importance in the cell. This is a complicated process, and so what we typically like to do anytime we have complicated processes is to break it up into sequences. I mean, that's what we did in cell respiration. We broke up this continuous process into glycolysis, Krebs cycle, electron transport. So for translation, we're going to break it up into 3 sequences, really just to allow our minds to kind of wrap up around the complexity of this. The first step is initiation, the second step is elongation, and the third step is termination. So basically start the polypeptide chain, lengthen the polypeptide chain, and then end that particular process. So what we're going to do is kind of go through each one of these particular phases and see what's involved. One of the things that I really want you to understand, because this is going to be important for upcoming discussions about regulation of gene expression is that this process of making proteins consumes a whole lot of energy. So one of the things I'm going to be pointing out to you is during initiation, where is ATP required? Or kind of an analog of ATP or a cousin of ATP, GTP. As far as you're concerned, you can think of these both as the same kind of substance. This is a form of energy that the cell can spend to do work. And this is work, to synthesize proteins. So keep this in mind and kind of be aware of that concept that translation of proteins consumes a lot of energy. Okay, so again, here's our kind of schematic of what is going to happen during the process of translation. We're going to have the interaction of a message, and specifically the code words on the message, with anticodons on the transfer RNAs. The ribosome is going to facilitate all these things coming together properly so that the code words are properly positioned. There's a place for the tRNAs to come in and bind so that they can read those code words. And then there are going to be some enzymes associated with these ribosomes. Sometimes they're RNA molecules, other times they're proteins, that will do things like form peptide bonds and hook these particular amino acids together, or slide the message through the ribosome so that new code words can be exposed and new information can be read in this particular message. So, let's start with initiation. The code word that starts the synthesis of proteins is always AUG. Okay? So this is a 3-base sequence in the messenger RNA. How do we know this is messenger RNA and not DNA? Uracil's present, okay? So this is the initiation codon in the message. The tRNA that will bind to that initiation codon, AUG, is the one that is complementary to it. And so it would have an anticodon of UAC. And the enzyme that charges up that tRNA with UAC will only stick the amino acid methionine onto that particular tRNA. So what does this ensure? This ensures that the message AUG basically gets turned into the amino acid methionine. Okay? This particular process requires a molecule of GTP, so there's some energy requirement there. And it also requires several proteins called initiation factors. That's as far as I'm going to make you go in terms of understanding the involvement of these substances, is that you need these extra proteins called initiation factors. If this were an advanced molecular biology course, you would probably have to learn about what those initiation factors do, what their names are, etc. But that's beyond the province of what we need to cover in this course right now. But it is important to understand that in addition to the requirement for energy, that this process requires other proteins to help it out. So, here's what it looks like. Sit tight. So here's our message. The first event that happens is that the small subunit of the ribosome, that would be the 40S subunit, positions itself on the message. There's a binding site here on the small subunit. And that the message gets attached to the small subunit, and then the initiator tRNA, the one that has the anticodon UAC, which is complementary to the AUG start codon, comes in and hydrogen bonds with that, and that carries the amino acid methionine. Now, this is a pretty tenuous relationship right here. What's holding that particular tRNA onto the start codon? It's just hydrogen bonding, right? Just hydrogen bonding between these A's and U's and G's and C's. And that could be broken pretty easily. So what immediately pretty much happens is once this comes together, the large subunit comes in there and docks in. And then that stabilizes everything. Okay? Remember that the ribosome has 3 docking sites. Well, actually 2 docking sites and an exit site. So this initiator tRNA is now positioned in the P site of the ribosome. The A site is still unoccupied, and the E site is empty at this point. And then this particular process requires a molecule of GTP. Okay, that's initiation. So now we're ready to go. What are we ready to do? We're ready to slide this message through the ribosome and expose new codons. And what's the next codon that's going to be read? This one that is sitting here in the A site. So what's going to have to happen? A tRNA is going to have to come in and bind to this A site. Which one? The one that has the anticodon that is complementary to this particular codon. And there are all those different tRNAs out there carrying different amino acids that might be trying to get into that docking site. But only the one that has the proper base pairing will go in there and stabilize. Okay? Okay, the next set of steps occurs over and over again as the next and the next and the next code word in the message are being read or interpreted. That particular part of the process is called the elongation cycle. This happens really fast. We can go through one complete elongation cycle in 60 milliseconds. Okay? That's really fast. That high rate of speed implies there are probably some enzymes that are involved in this process that help to facilitate this particular reaction. So, during elongation, a messenger RNA codon is read, and then the ribosome or the message move in relationship to one another, and the next codon is read. That process of moving down the message is called translocation. Every time a new tRNA comes into the A site, you can think of the A site as the acceptor site, the one that's going to bring in a new tRNA. Each time that happens, it requires a molecule of GTP, so more energy. Movement of the message and the ribosome relative to one another also requires energy. And that kind of makes intuitive sense. If things are sliding past one another, we're probably going to be consuming energy. And one thing I didn't put on here, I probably should have, if you're taking notes, this process of elongation also requires other proteins. They are called elongation factors. So just as the process of initiation requires some additional proteins, initiation factors, this process of elongation requires some other proteins as well. Okay, so here is what a typical elongation cycle looks like. So let's just kind of take it from here, and we will go through one elongation cycle, 60 milliseconds worth of time. Okay? Now, this shows you a polypeptide chain that has already been through several elongation cycles. How many elongation cycles has this already been through? It's been through 3, right? It's been through one initiation, and then 1, 2, 3 elongation cycles. So it's getting ready to start the next one. So it's read some code words in this message already. It's getting ready to read the next one. Okay? The A site is unoccupied. And at any given time, a ribosome can bind 2 tRNAs. Right now it's only got 1 bound. So what happens? Well, this A site is empty. So all these different tRNAs are going to try to get in and dock at that A site. But the only one that can do that is the one that has an anticodon that is complementary to this particular codon right here. Okay? That particular docking and positioning requires energy. So there's more energy that is being used up. Okay? Once both of these sites are filled now, an enzyme gets activated, and that enzyme is part of the structure of the ribosome. What does that enzyme do? It catalyzes the formation of a peptide bond between this amino acid and this short polypeptide chain. So now a bond is forming here, and this bond becomes broken. So notice the outcome here. Now all these amino acids, which were previously on the tRNA and the P site, are now attached to the tRNA and the A site. This tRNA no longer has amino acid. Its amino acid has been transferred over here. So what happens? Well, this message now slides through the ribosome. So now, this particular transfer RNA, which was in the A site, is now moved over to the P site. This one that was in the P site is in the E site and is ejected or exits. And now a new A site is open. Okay? This particular codon is now exposed in the new A site. And what happens? We have another elongation event again. And another. And another. And another. This is the one that sometimes gives students conceptual problem, is the sliding of these messages and how that moves that tRNA from one side to another. And that's where I think some of those animations, we'll show you an animation in a second, but I think some of those kind of real-time animations help you to visualize that a little bit better. So the message is kind of moving this direction, but effectively what's happening then is that message is being read in this direction, in the 5' to 3' direction. Okay, that's going to just continue until we reach a stop codon. And when we reach a stop codon, that is going to signal termination of protein synthesis. Basically, when that stop codon is reached, everything falls apart. The message releases from the ribosomes separate into their two subunits again, and the polypeptide chain is released so it can then fold up, and once it's folded up in the proper shape, it does some job in the cell. Now, important thing here, when you reach that termination codon, remember there are 3 stop codons or 3 termination codons in the genetic code. There's not a tRNA that corresponds to that termination codon. Remember we said that there were only 61 code words, even though there are 64 possible combinations. So what happens? Well, instead of a tRNA moving into that P site, that A site, sorry, some proteins bind to that A site. And those proteins are called release factors. And that's basically what causes everything to fall apart and the process to stop. And then polypeptide chain folds up. So, here's our stop codon, 3 stop codons. Here's our tRNA that has been translocated over into the P site. Here's all of the amino acids that have been strung together by reading these code words. Oops. But now we hit this stop codon. No tRNA that is complementary to that particular stop codon. But these proteins, release factors, come in and bind there instead. This bond is broken, polypeptide chain is released. This tRNA is released. The ribosomal subunits separate, and the message is released. Could that message potentially go back now and reassociate with the small subunit, position itself at the initiation codon, and be read again? Sure. Why not? Will that happen forever? No. Every molecule in the cell has some kind of a limited lifetime. And eventually molecules are degraded. Do you remember what we called that process back at the beginning of the course? It's called turnover. So molecules turn over, are broken down, and then they have to be resynthesized again. Okay? That actually is important that the cell has turnover, and will be important when we talk about regulation of gene expression. Because if there wasn't turnover and these molecules lasted forever, then once a message was made, it would always be transcribed. That gene would always be expressed. And you might not want it expressed all the time. Okay, so here's a rough estimate of what you need to make a protein molecule. The rough estimate is that for every amino acid that's incorporated into a polypeptide chain, about 3 ATP/GTP are required. One for charging the amino acid, one for binding the incoming tRNA to the A site, one for translocation. So what's the implications of that? Well, if you have a protein of 120 amino acids, which is a pretty small protein, a lot of proteins are thousands of amino acids in one. But if you have this really small protein, if it takes approximately 3 ATPs per amino acid, that's 360 ATPs to make that molecule. What's that the equivalent of? That's the equivalent of about 12 glucose molecules going through aerobic cellular respiration. So it takes a lot of food to make these proteins. That's why when we talk about regulation of gene expression, we will see that organisms have evolved mechanisms to only produce proteins when they need them. Why burn up your sugar, your energy source, making a protein you don't need? So that's really going to be important. Another nifty mechanism that the cells have evolved over time to kind of ramp up their production of proteins is polyribosomes. It takes about a minute for a single ribosome to translate an average size polypeptide chain. What if you really need to make a lot of that protein? Well, in reality, what can happen is several ribosomes can translate the same message one after another. Think of it as a train on tracks. And think of translation as the train moving down those tracks. You have to wait till one train gets to the complete end of the track before another one can start going down. No. Those trains or ribosomes can just line up behind one another and start chugging down the track right behind one another, making the polypeptide chain. So here are multiple ribosomes, polyribosomes, attached to the same message. This one is just finished translating the message. It's hit the stop codon. And now it's made it its complete polypeptide chain. This one is almost done. This one is partly done. This one is kind of just started. And then here's some more incoming ribosomal subunits that are just getting ready to begin the process of protein synthesis. This is an actual electron micrograph that shows polyribosomes, all these ribosomes lined up on the same message, moving down the message simultaneously, forming multiple copies of that particular protein. One other little wrinkle in this that I want to mention. Do you remember the difference between secreted proteins and proteins that stay in the cytoplasm of the cell? What's the difference between proteins that are secreted and proteins that stay in the cytoplasm? Back to about the third week of class. Has to do with the ribosomes that they're made on, right? What kind of ribosomes are secreted proteins produced on?
Classic.
Yeah, membrane bound, often the ER. And the proteins that stay in the cytoplasm are made on, remember the designation? Free ribosomes, the ones that aren't membrane bound. So, for example, the enzymes of glycolysis, do we want to secrete those? No, they need to stay in the cell where they're produced, because that's where the energy is produced. But what about the hormone insulin? Insulin's a polypeptide chain. That's got to be secreted, right? It's got to go out into the circulatory system, and then it can bind to target cells in the liver and the muscles and help regulate your blood glucose levels. Okay? Well, as it turns out, all proteins start being synthesized initially on 3 ribosomes. But at some point, the ones that are secreted, the ribosome hooks up with the ER membrane, and that starts getting pushed into the channels of the ER and is eventually exported out of the cell. So a couple of questions. Number one, what causes some of these ribosomes to hook up to the ER, but not other ones? Another way of saying that is, why are some proteins secreted and others aren't? Well, that information is actually encoded into the messenger RNA. And encoded into the message is something called a signal sequence. And basically the signal sequence provides the information, is this going to be secreted or not? If it's going to be secreted, the ribosome and message are eventually going to hook up to the ER, and that's going to push the protein out of the cell. If the protein's not destined to be secreted, then there will be a different signal sequence, and that ribosome will never hook up to an ER membrane. So, here's what it looks like. Here we've got a message, here we've got the ribosome. And as it turns out, this particular message encodes a protein that eventually the cell is going to want to secrete. So there's a series of nucleotides in the DNA that say secrete. And those are copied by transcription into an RNA sequence that's part of the message. And that codes for something called a signal peptide. That signal peptide basically says, ribosome, hook up to this ER and push this protein out of the cell. So secreted proteins have a signal sequence, non-secreted proteins don't. Okay? So that's genetically encoded. Now, it's a little bit more complicated than that because there is another protein called SRP. SRP stands for signal recognition particle. And this little molecule recognizes the signal sequence. If there's no signal peptide being produced here, SRP won't bind. This concept of SRP and signal peptide binds to a little receptor on the ER. If the complex doesn't form, this isn't going to bind to the receptor. If it binds to the receptor, then the ribosome gets held there, and that protein now starts snaking into the ER channels. Eventually that signal peptide is removed, and then once this synthesis is completed, this protein then moves through the ER channels and eventually might go to the Golgi for modification, but eventually gets packaged up in a vesicle and gets secreted by the cell by exocytosis. Okay? So this helps to answer one of those questions about how proteins know where to go. Why are some secreted, some not secreted? because it's encoded actually into the instructions for that particular protein. Okay? Okay, let's try a couple of videos here. Let me get this to work. Whoops.
Messenger RNA is used to direct the synthesis of a polypeptide. This information is carried in the sequence of bases on the messenger RNA molecule. The polypeptide can be assembled once RNA binds to a ribosome. Ribosomes consist of large and small subunits. The large subunit has two binding sites for transfer RNA, the P and A sites. Transfer RNA carries an amino acid, which will be incorporated into the polypeptide. Its anticodon is a triplet complementary to the codon on messenger RNA that specifies.
Now, do you see how important it is that the correct amino acid get attached to the correct tRNA with a particular anticodon? Because we want to ensure that when this anticodon interacts with the message is saying methionine, okay? So we don't want tryptophan to get on this particular amino acid, which is reading that code word that should say methionine. Otherwise, we're going to make the wrong protein. We're going to have the wrong primary structure, and it's going to fold up improperly. So these enzymes that put together the proper tRNA with the proper amino acid are really critical for making this genetic coding system work the way it does.
That particular amino acid. A ribosome assembles on the start codon AUG. Transfer RNA with the anticodon UAC and carrying the amino acid methionine binds to the codon. The transfer RNA is in the P site of the ribosome. The A site is available for a second transfer RNA with an anticodon complementary to the second messenger RNA triplet. The amino acid carried by the second transfer RNA binds to the methionine. The first transfer RNA leaves the P site, and the second transfer RNA moves there, still bound to messenger RNA.
So you see how they're showing the message sliding through the ribosome, and that's probably the correct way to interpret that. But by this sliding this way, this ribosome is effectively moving this direction. Does that make sense? So we're really reading this message in the 5' to 3' direction because this message slides through the ribosome in this direction. That can be a little confusing sometimes.
This brings the third messenger RNA codon to the now empty A site, and the appropriate transfer RNA can bind to it. The third amino acid is added to the chain, and translocation occurs once again. This process of polypeptide chain elongation continues until a stop codon is reached. A release factor binds to the A site. It carries no amino acid, but facilitates release of the polypeptide and the messenger RNA from the ribosome.
Are you going to make a comment, Larry? Or?
No.
Okay. I'm going to show you one more, and then we'll do another one on Thursday, but you need a little bit more background for that one. Let's take a look at this one for a second.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule.
So when they say molecular machine now, they're talking about RNA polymerase, okay, the enzyme.
Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins.
There's our charged tRNAs. Codon,
Inside the
anticodon.
ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured.
You can see empty tRNAs or tRNAs that no longer have an amino acid attached being spit out of that E site. And then you can see the polypeptide chain being assembled as translation is happening.
When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
Now, we didn't talk yet about the, and probably won't, about the involvement of this barrel-shaped molecular machine that helps to facilitate folding up of the proteins. That's a little beyond our understanding.
Oh, but it's got a great name. Chaperonin.
Chaperonin, yes. Okay, there's a couple of other wrinkles that you're going to have to be made aware of that have to do with the messenger RNA in eukaryotic cells only. So this is going to be relevant to your messenger RNAs, but not bacterial messenger RNAs, for example. Once the messenger RNA is transcribed after transcription, there are three modifications that typically happen to eukaryotic messenger RNA. Where does transcription happen? Where in the cell? Nucleus or cytoplasm? Nucleus, okay? So these are going to happen in the nucleus before that message then moves through the nuclear pores and goes into the cytoplasm and hooks up with the ribosomes, okay? The 3' end of the message is modified by having something called a poly-A tail added onto it. What is a poly-A tail? It's a series of just AAAAs. Why is it doing that? Well, we'll have to talk about that. Okay? The 5' end, the other end, is also modified by having a modified guanine nucleotide stuck onto the 5' end. That's called the cap. And then finally, some segments of the message are actually cut out. That seems weird because if you cut out nucleotides from the message, aren't you losing genetic information to make a polypeptide chain? Well, you would think so, but as it turns out, you aren't. These particular sequences are called introns. And basically, if the message has introns, the genetic information in that message is split up. And it's separated by non-coding spacers. When we say non-coding, we mean that those spacers aren't specifying amino acid sequences. So in the DNA, there are actually something in eukaryotic cells called split genes, where if we've got this segment of DNA, there are regions that are coding for amino acid sequences called exons. But then they're separated from one another by non-coding sequences called introns. And then we might have another exon here. And when a message is formed, this all is copied. So the message is also going to have exon 1, exon 2, intron 1, exon 3. Well, that seems really strange. This was a very interesting discovery when they figured out that this is indeed the case. If you tried to translate this message with the introns in it, it would screw it all up. So before translation occurs, this intron's got to be chopped out. And then all the exons are joined together. That's bizarre. And that represents the potential problems for the cell. So what if this isn't cut out properly, for example? What if you make a mistake cutting this out? And why do you go to the problem of copying something, transcribing it, and then just cut it out? So those are three things that we will talk about. Actually, Dr. Morton will talk about on Thursday. Dr. Morton McCombs and I have to go to a life sciences breakfast in Indianapolis, so Dr. Morton's going to fill in for me on Thursday. And he's going to be talking about some of these modifications to messenger RNA. And then, time permitting, he will also introduce and talk about what happens if your DNA changes. What happens if there are errors in the DNA mutation. And that is very timely because you're seeing a video in lab this week called Accidents of Creation, which talks about the role that mutation plays in evolution. Okay? So, we get a little bit of a break today. This is a good ending point.